Good morning. Topic for the day is antigens. What are antigens? Antigens are substances that can stimulate immune response. For a substance to be antigenic, it should satisfy two immunologic properties, that is, immunogenicity and antigenicity. When B cells or T cells interact with antigens, they form an effector B cell or a T cell that contribute to immune response. Antigenicity is the ability of an antigen to combine specifically with the final product of above two responses. All molecules having immunogenic property also show antigenicity, but reverse is always not true. Complete antigens that induce antibody formation and produce a specific reaction with the antibody so produced. Epitope or antigenic determinants are the smallest unit of antigenicity. Epitope is the small area present on the antigen that is capable of sensitizing T and B cells and reacting with specific site of T cell receptors on an antibody and specific site of an antibody that react with the corresponding epitope of an antigen is known as a paratope. Haptens. Haptens are low molecular weight molecules which cannot induce an immune response, but they retain antigenicity or immunologic reactivity. They can bind with specific antibody or T cell receptors. They can become immunogenic when combined with a larger protein molecule called carrier molecule. Hapten carrier complex is capable of inducing immune response in the body. Haptens are classified as complex haptens and simple haptens based on the number of epitopes. Complex haptens contain two or more epitopes and can react with specific antibody, whereas simple haptens contain only one epitope. Based on the antigen or host relationship, self or outer antigens and non-self antigens are there. Self antigens belong to the host itself, but they are not immunogenic. Host immune system does not react to its own antigen due to a mechanism called immunological tolerance. And foreign antigens or non-self antigens are immunogenic. And based on this property, antigens are classified into alloantigen. That is, they are species specific. Tissues of all individuals in a species contain species specific antigen. Second one, isoantigens. It is present only in the subset of a species. It is unique to every individual of a species. An example is the blood group antigen and histocompatibility antigen. And last one, heteroantigens. Heteroantigens belongs to two different species. That is antigen of a plant or an animal or a microorganism. Heterophile antigens are a type of heteroantigen that is present in two different species, but they share epitopes with each other. That is, antibody produced against antigen of one species can react with other and vice versa. Adjuvants. Adjuvants are substances that enhance the immunogenicity of an antigen. They are usually added to vaccine to increase the immunogenicity of vaccine antigen. Examples of adjuvant activity are alum and mineral oil. Lipopolysaccharide fraction of gram-negative bacilli also serve as an excellent adjuvant. LPS of Bordetella petrosis is an excellent adjuvant for diphtheria and tetanus toxoids. Biological classes of antigens. Based on the mechanism of inducing antibody formation, there are two types of antigen. They are T cell dependent antigens and T cell independent antigens. Normal antigens are usually T dependent. They are processed and presented by antigen presenting cell to T cells, which leads to T cell activation. The activated T cell secretes cytokines that in turn stimulate B cells to produce antibodies. T cell independent antigen. Few antigens such as bacterial capsule, flagella, and LPS that do not need the help of T cells and antigen presenting cells. They directly bind to immunoglobulin receptor present on B cells and stimulate B cells polyclonally. This results in the secretion of a large amount of non-specific antibodies, and the condition is known as hypergamma globulinemia. TI antigen can activate both mature and immature B cells, but B cell can differentiate into activated cell and no memory cell formation is seen. Superantigens are yet another classes of antigen that can activate T cell directly without being processed by antigen presenting cells. The variable region of T cell receptor appears to the receptor for superantigen. They directly bridge non-specifically between MHC class II of APCs and T cell. The action of superantigen results in the release of T cells, that is, non-specific activation of T cells, massive release of cytokines, activates B cells polyclonally, and increased secretion of non-specific antibodies. All these conditions result in various clinical manifestations. Examples of superantigens are staphylococcal toxin and streptococcal pyogenic exotoxin. Viral superantigens include CMV-associated superantigen, rabies nucleocapsid, and HIV-encoded superantigen. Malassezia focus is an example of a superantigen. And examples of some diseases associated with superantigens are toxic shock syndrome, food poisoning, scalded skin syndrome, and rare conditions like atopic dermatitis, Kawasaki syndrome, psoriasis, acute disseminated encephalomyelitis, etc. This is a pictorial representation showing the action of superantigen trespassing MHC class II. There are various factors affecting the immunogenic property of an antigen. First one is the size of the antigen. Larger the size, more potent is the molecule as immunogen. Chemical nature of the antigen. Proteins are stronger immunogens than carbohydrates, and they followed by lipids and nucleic acids. Susceptibility of antigen to tissue enzyme. Substances that can be metabolized or are susceptible to the action of tissue enzymes are immunogenic. And foreignness to the host is the key factor which determines immunogenicity. Non-self antigens induce an immune response as the host distinguishes self from non-self and does not respond to self. Antigens from other individuals of same species are less antigenic than those from other species. Genetic factor due to genetic differences between the individuals. And finally, optimal dose of antigen. A too little dose of antigen elicit immune response, and too large dose lead to the development of immunological tolerance. And that's all for antigens.